Okay, so I will begin now. So thank you everyone for coming to my defense. So the outline for today is some basic introduction, and then I'll go over the 3 main projects that I did for my PhD. The first 2 are already published, so I will just go over their main ideas, and I will concentrate on this final project. So one of the most important topics discussed in this dissertation is Genome-Wide Association Studies, so GWAS. And in GWAS, we want to study the genetic basis of some phenotype, okay, like height or whether you develop cancer, right? It could be any phenotype. And what you do in GWAS is you recruit N samples, and then you genotype these samples at P SNPs. So these are just genotypes of your entire genome. And then you can assemble this matrix that is N by P. Every row is a sample. Every column is a SNP or a genotype. And the key is usually N is about from thousands to like 100,000s, but P is usually maybe like 1,000,000. So this is a high dimensional problem, right? And so it's that's partly what makes it a little bit hard. But anyways, you have this matrix, and then you also measure each sample's phenotype. So you get this vector, and you want to ask which SNPs are associated with the phenotype. Okay, so which columns of X is correlated with Y in some sense. Okay, so that's what GWAS is. And because GWAS is very popular for the past decade, there are lots of methods developed for GWAS. Here I broadly categorize them into two main categories. The first one is the sparse models, okay, the sparse penalized regression models. In this regime, you estimate the effect of all SNPs jointly in a single model. And the assumption is that even though you include all of the SNPs, only a few of them are truly causal for your phenotype or associated with your phenotype. Okay, so almost every other SNP have 0 effect. And if this is your model, you usually run penalized regression with penalties such as the IHT penalty. And that's the main thing we will focus on. But there are other penalties like the lasso, MCP, or Bayesian analogs of these guys. So that's the sparse model. And then on the other hand, there's the linear mixed models, okay? And in this regime, we focus on the effect of a single SNP at a time. And the assumption is each of those SNPs will have normally distributed effect size. And if this is your model, you basically do hypothesis testing, okay? And the linear mixed models is implemented in lots of software that are very highly cited. So what we want to say in this thesis is the relative performance of these two regimes, these two kind of modeling framework, they sort of vary depending on the true genetic architecture of your phenotype, right? And no method is uniformly superior for all methods. So in some sense, they sort of complement each other rather than compete, right? And at least that's what we're trying to say, okay? So now we know what GWAS is. I want to go over a few questions that are discussed in this overall dissertation. They're all motivated by GWAS, but they might not be directly related, right? For instance, phenotypes Y. So this can be like binary, right? Or it could be continuous, it could be count data. So whatever statistical model you developed has to respect that structure, right? You can't have 2.5 seeds per plant, right? So how do you respect the structure of Y? How do you deal with missing genotypes, right? GWAS samples 1 million SNPs, but there are 3 billion base pairs in the human genome, right? So can you impute them? Can you impute the rest? Is there meaning in doing that? Population structure is a major confounder. How do you deal with that? How do you even compute with these big data of big Y, big X? If they're so big, it's not trivial to deal with them. And what do we do when we have multiple phenotypes, right? So these will be discussed in the next 1 hour. So now I will move on to the first project. It is extending the iterative hard thresholding in 3 ways, okay? But we will only focus on one of them. So the paper is published in Giga Science, and accompanying software and documentation is available here. So first, let us consider the structure of Y, right? So if Y is binary or if it's count, how do you deal with that? We will make 2 assumptions. The first is samples are independent, okay? And the second assumption is that the traits follow some exponential family of distribution. So if you assume these 2 assumptions, then you can model Y as a generalized linear model, okay? And this includes the case of binary or count or continuous, right? So it covers many cases. And the idea is, even though Y is, it's binary or count, it has a special structure, the expected value of Y is continuous. And you can model that as a nonlinear transformation of your linear covariates, okay? And the reason we want to do general linear models is because the log likelihood can be expressed in a very general way, like this. And from this, you can compute the gradient. And from the gradient, you can compute the Hessian, and from the Hessian, the expected information matrix. Okay. So once you have these ingredients, you are ready to do IHT on high-dimensional generalized linear models. Okay, so here's the problem, right? How do you do IHT on GLMs? We will maximize the log likelihood subject to the sparsity constraint, right? The number of non-zero entries of beta is less than k. And this is how do you solve this with IHT? You basically repeatedly iterate 3 core ideas until it converges, which is you start with your current estimate of beta, you compute the gradient, which is given here. You compute a step size, it's given here, and this includes a quadratic form of your expected information matrix. Once you do that, so basically a gradient step, right? You take that resulting vector and then you project it to sparsity. So that involves finding the k largest entry in magnitude with a partial sort, and then you set everything else to 0, right? So just gradient step followed by projection, and then you keep doing that until convergence. So this is sort of one of the main results of our first paper. We are comparing IHT to Lasso. This is the most popular sparse regression model. And then we compared also to the marginal testing, so the SNP by SNP p-value method. This is on simulated data. I guess the summary is that comparing IHT to Lasso has a lot of false positives. Okay, so this is too much to follow up on in terms of GWAS. And comparing IHT to the marginal, the marginals have much better controlled false positive rates, as they should, but the true positive rate is lower. Okay, so that's the summary. And then to illustrate the scalability, we applied IHT to the UK Biobank. Okay, and we will actually reanalyze this data in my 3rd project. So there I will go over more of the computational and pre-data process cleaning. So here's just really just some preview material, I guess. But anyways, the UK Biobank data here contains 200,000 samples and 500,000 SNPs. The phenotype is blood pressure, okay? So it's some way you can measure the blood pressure and you can dichotomize them so that you can categorize people whether they have hypertension or not. So we ran at IHT, and we also ran a traditional univariate, SNP-by-SNP p-value. And if you compute the p-values, you can put them on this Manhattan plot. The higher the dots, the more significant the SNP. And then the circle dots are what is found by IHT. Okay, so I guess the summary is IHT typically finds these large signals from a traditional analysis, but IHT also finds a lot of these SNPs on the bottom. Okay, and if you believe our simulation here, right, basically for the logistic case, for binary case, IHT has very little false positive, right? So probably a few of these they might be real. Okay. And they are real in the sense that traditional methods cannot detect them. So anyways, that was the...
Then here, the hypertension is the binary outcome?
Yeah, we categorize we measure the systolic, diastolic blood pressure, and then we can categorize them into samples, whether they have hypertension or not.
Mm-hmm.
It's just to illustrate we can do logistic GWAS.
So for the signals which the marginal p-value actually is not significant at all, did you follow up, like are they in some meaningful genes or something?
I didn't follow up.
You did not?
Yeah, I think it would be good if we can run gens analysis on these guys. I don't know.
I see, yeah.
So now I guess we will go on to the second project, unless there are more questions. So the second project is about imputation, genotype imputation and phasing, and ancestry inference. Okay, the paper was published in Bioinformatics, and the software that accompanies it is here, and the documentation is here. So genotype imputation, right? What is it? What is the goal? So first of all, the main goal is you can increase the number of markers in the GWAS. Right, you have 1 million SNPs, you can impute them, and then suddenly you get 10 million or 100 million SNPs. Okay. And another goal is to do meta-analysis, right? If you have multiple scientists, they each do their own GWAS on different sets of 1 million SNPs, and you want to combine them, right? How do you do that? You can just impute all of those data to the same panel, and then they will all be on the same set of SNPs. So you can just do standard GWAS, right? So in genotype imputation, there's two inputs. One is your GWAS data. So it's visualized here. Every row is a sample, right? Every column is a SNP. And then the second input is a reference panel. Okay, so it's visualized here. There's many, many more SNP in this reference panel, but it also is phased. Okay, so what that means is every 2 rows is 1 sample. And these 2 rows are the haplotypes of the samples, right? So each genotype vector can be decomposed as the sum of 2 haplotypes. So reference haplotypes and GWAS data here, you combine them, and what you want is you want to, first of all, impute all of the missing data, and you want to phase all of them, all of the samples. So that's the process of imputation and phasing. So there's this online service called the Michigan's Imputation Server. And basically you can upload your data there and then they will impute it for you and you can download it. They impute 10 million genomes every year, right? So it really, genotype imputation is becoming a standard procedure in genetics nowadays. And because of that, the reference panels that are available, they have been getting extremely big, right? Every 3 or 4 years, a major one comes out, and that will be 30 times bigger than the previous biggest panel. Okay, so probably in 2022, 2023, we will see another one. I don't know. But anyways, the methods that will run with these reference panels, they are all based on hidden Markov models. Okay, so for instance, Minimac, Beagle, and Impute, these are the main imputation softwares. And in fact, they are all based on the same hidden Markov model. Okay, it's the Lee and Stevens HMM derived in here. And because of that, all of these softwares have very similar imputation accuracy. And they also require you to pre-phase their data. So what that means is here, the input genotypes, they have to be phased first. And that phasing is also non-trivial. In fact, sometimes it takes longer than imputation, okay? So these are not the main issues we have, right? The main issue that we are concerned with is that HMM methods are not very fast, okay? So with reference panels growing so fast, there is probably some value in developing a very fast imputation algorithm. And that's what we try to do. Here's a case study, just to illustrate that HMM methods are actually slow, right? I ran Minimac 4 on a local computer. So by the way, Minimac 4 is used on Michigan server, right? So almost everyone is using Minimac 4. If you impute 1,000 samples using the HRC panel, that's the 2016 panel, this problem will take about 3 hours on one chromosome, okay? So if you want to impute every chromosome on every sample in the UK Biobank, this would probably take you about 4 years, okay, rough estimate. And if you have to do pre-phasing, then you double this. And then if you want to run with, the latest panel, the top met panel, then you multiply that by 30, okay? So it really is a very slow process. And here's just advertisements for our own software. If you use our software, this time will be approximately 20 days. It will not require you to do pre-phasing. And you will also get Ancestry data automatically at the end of imputation. Okay, so you get it for free. Our software also supports a bunch of different input formats. The VCF is the main one, but there are other ones. So I guess that's the background. In the next 2 slides, I will go over how we do imputation. Basically, we have to do imputation and we have to do phasing, right? So it's a 2-step process. First, we will do imputation. Let's look at a small genomic window. Okay, so genotype vector for a sample, this vector length is like maybe 100 SNP, okay? Entries is 0, 1, or 2, or missing. And then because you're looking at a small window, the number of reference haplotypes, you can filter them for uniqueness. You only want to look at the unique ones. And usually there are about 1,000 unique haplotypes per window. So given this setup, if you want to do imputation, you basically want to find 2 haplotypes such that their vector sum is approximately X, right? And that can be done by minimizing this least squares problem over all possible HI and HJ. So how do we do this? The idea is basically we will collect these terms involving inner products of H's into a matrix, M, and then these terms involving inner products of X and H's into another matrix. And these 2 matrices can be very quickly assembled from matrix-matrix multiplications. We invoke BLAST3 to compute these, so they are very fast. And then once you have these 2 matrices, you ignore this first term because it's not involved in optimization. And then you can search for the minimum entry of this sum, this symmetric matrix. And searching that will minimize this criterion. Okay. And the key is, because we're looking at genomic windows, we can basically parallelize this process by having each CPU core handle a different window, right? So that's how you do imputation. And then the next step is phasing, okay? So once you did imputation, right, each window you found 2 haplotypes that are the best for that window, right? So for instance, H1 and H5. And remember, these haplotypes are unique, right? So there are multiple other haplotypes that are equivalent to the unique one. So this is visualized here. And what you want to do now is you want to basically have these long haplotype stretches to stretch as long as possible, but allowing for a switch of the 2 sets in a window. And you want to switch them if that will give you a longer haplotype stretch. Okay, so for instance here, H5 survives for 3 windows. But if you do a switch here, it will allow H5 to continue to survive. And if switching doesn't help, so here, H1, you can now find an H1 here, then you eventually have to search for a breakpoint in the middle, okay? So in our software, we first impute and then we phase, okay? And that's contrary to HMM methods, which phase and then impute, right? So I guess it's kind of different. So let's look at our result. Okay, I compared our software, Mendel Impute, to the 3 latest HMM software. Here's the 1000 genomes panel, and here is using the HRC panel. Okay, the summary is that we are a lot faster. Okay, we are usually 10 or even 100 times faster than these methods, and we require much less RAM, much less computer memory to do these computations, because we are working with small genomic windows. But we do pay a price, which is that our imputation accuracy is slightly worse than these other methods. We are 1.5 to 2 times worse than they are. But even then, our error rate is still quite low, right? You're looking at maybe 1 imputation error out of every 1,000 steps. So it's still a pretty good error rate. So finally, we also did, so I said, we can get Ancestry data for free, right? Here is one of the Ancestry results. Basically, every sample is a column. And then what I'm doing is I'm computing the proportion of ancestries for each sample, right? So here, this sample here is maybe 70% African, 20% European, and 5% South or East Asian. East Asian here represent Amerindian population, so native Indian population. So how do we get this? How do we get this information, right, from imputation? It comes from phasing, right? Remember here when I did the phasing, I'm saying H1, H5, but these haplotypes, they are from the reference panel, right? And usually you know something about the reference panel, right? For instance, if H5 is a European haplotype, then you know that at least across these 4 windows, this person is at least 50% European, right? So you can calculate that in this process. And that's exactly what we did here. So we took our output and then we compared it to this software, Admixture. It is the main software used for this kind of analysis. So if you just look at these pictures broadly, the summary is that for the Colombians, the Mexicans, and the Puerto Ricans, the 2 results are very similar, right? But for the African populations here, it turns out that Mendel Impute were more accurate than Admixture, because we know that the African Americans, they tend to have about 15–20% European ancestry. And for some reason, Admixture thinks they are about 30% Amerindian. So that's just, I don't know, that's not true, right? But also for here, for the Peruvians, it turns out the admixture was a lot more accurate because Peruvians do have about 50 to 90% Amerindian. So why is there a discrepancy? It requires more studies. This is just an illustration. So that was the final result. Is there any questions? Okay, so finally, I will move on to the final project that we worked on. It is an extension of my first project on IHT. It's still under review, but you can see our paper on arXiv, and the software and documentation is here. So we will revisit GWAS, right? And the question is, what do you do in GWAS if you have multiple phenotypes? Okay. So the stupid thing that you can do is you can just do a separate GWAS for each of those phenotypes. Okay. And that implicitly assumes phenotypes are independent. And the problem, of course, is that phenotypes are correlated, right? If you have high BMI, then probably you have higher blood pressure. So in fact, there are multiple studies that analyzed the difference between multiple univariate GWAS versus a multivariate GWAS. And they basically summarize their main advantages of multivariate GWAS here, right? Multivariate, when you model all the phenotypes together, you have more information on the correlation between phenotypes. You can distinguish between independent and pleiotropic SNPs. So pleiotropic means a SNP affects multiple phenotypes. It reduces multiple testing burden, and in fact, multivariate GWAS has higher power than univariate ones. And what is surprising is that these advantages are true even if there is very weak correlation among the phenotypes, or even if one of the phenotype is like simulated, okay, it's completely unrelated to your genotypes, these advantages still hold. So these reviews, they almost always conclude by saying you should just do multivariate GWAS as long as it is computationally feasible, right? And of course, this is one of the problems, okay? So in a lot of the cases, it's not feasible. So that's one of our main goals of this project, is to have a very fast software for multivariate GWAS. So let's look at existing methods for multivariate GWAS. There's a software called Gemma. It implements a multivariate version of linear mixed model. So probably you should use this if you want to do multivariate GWAS. There's another one called multivariate Plink. It's much older, but it is very fast. It is the second fastest method found in the review. And we will compare against these two methods with IHT later, okay? So Gemma for statistic performance and Plink for speed, okay? And by the way, we did not compare against the fastest method because the fastest method was an older version of Gemma. It implements some Bayesian method. Okay. And there, of course, there are a bunch of other ones. Okay. But it turns out that there are no methods. All of these methods considers the SNP by SNP association model, right? There are no method that does penalized regression. So I think it would be one of our other second main contribution is we present a penalized regression method for multivariate GWAS. So how are we going to do this? We will, again, make two assumptions. The first is the independent sample assumption, right? So it's the same as the univariate case. And the second assumption is that each of those phenotypes, there are different phenotypes, they have normal distribution. Okay, and basically we will model each phenotype vector for each sample as a multivariate normal. Okay, and because samples are independent, their likelihoods multiply, right? So overall, you have this log likelihood. Gamma is the inverse covariance matrix, right? It's usually in the middle, but you can pull it out in front using the trace trick. So what we will do is we will maximize the log likelihood with two constraints. The first is the sparsity constraint, right? The regression coefficient matrix B has to be k-sparse, okay? And the precision matrix gamma, it has to be symmetric and positive definite because it's the inverse of a symmetric and positive definite matrix. And how are we going to do that, right? Of course, we're going to use IHT. So optimizing for the regression coefficient is basically the same as the univariate case, where you take your current B, you compute the gradient, it's given here, and then you compute a step size, it's given here, and it has a slightly more complicated quadratic form. And then you take that resulting matrix and you project it to sparsity, okay, finding the k largest entry, and then setting everything else to 0. Okay, so you do one IHT update, and then you do an update for gamma, okay? And this update equation is motivated by the stationary condition of the log likelihood with respect, the gradient with respect to the gamma. So what we'll do is we will run one iteration of B and then use that to update gamma and then repeat them until convergence. There is this matrix inverse here. And this matrix is R by R, okay? R is the number of traits. So as long as you're not doing like 100,000 traits, this, if you're doing 100 traits or 1,000 traits, this inversion is trivial, okay? So now let's talk about the performance step, okay? So it turns out that in IHT, the most computationally intensive step is gradient calculation, okay? So calculating gradient. And that involves doing a matrix or a matrix vector multiplication. Here A is your genotypes, and B and C are numeric matrices. And the idea is, because A can be super big, right? It could be 500,000 by 500,000. You don't want to do it the naive way. So you want to keep them compressed to save memory. But at the same time, you have to do this fast, right? So how do we do that? What we do is we employ this blocking strategy, okay, or the tiling strategy. So it takes this representation of C, okay, so you can cut C into smaller blocks, and then each of these smaller blocks can be computed by this tiled product. So the advantage of doing this is that because these smaller blocks of A and B, they are small, accessing smaller blocks will improve cache efficiency. And these blocks can be multiplied in parallel. And most importantly, each of these small product, we can vectorize them using the loop vectorization package in Julia. And that will basically calling SIMD on this product. And that will, this is actually the most significant speed gain from this entire thing. And if you do this strategy, the performance is that we can do this matrix multiplication as fast as a decompressed numeric matrix if B is tall and thin, or flat, short and fat here. So it is as fast as +3 if B is not crazy, okay? And in fact, if B is a vector, so in the univariate IHT case, the matrix vector multiplication for this routine is actually usually 2 times as fast as +2, okay? And this speed gain, the speed is predicated on a compressed genotype matrix A, okay? So A is 32 times smaller than a decompressed matrix. So this is for binary Plink files, all of these strategies. If you have other inputs like VCF and bgen, we will just import them into numeric matrix and then you use BOS.
I have a technical question. Does the loop vectorization package work for all the CPU architecture? Like ARM and AMD, does this work for those CPUs?
I'm not sure. Sorry.
Does this require CPU to have certain instruction sets or something?
I think
you run on Intel CPU.
Yeah, in the early days, I think they said they need AVX machine. But at some point, I think they dropped that in their documentation. I'm not sure. I never tested it though.
So you ran on Intel machines or?
Yes, yes.
Okay, I see. Seyong, do you know? I just want curious.
I haven't tested, but I think it's targeting more general hardware now.
So like ARM architecture can also work. I see.
Yeah, I think so.
Okay, thank you.
So now we will go into, we will test IHD on simulated data. We used the NFBC, the Northern Finland birth cohort data for simulation. We will simulate the phenotypes from a matrix normal distribution. And this is actually the model adapted by the linear mixed models. Okay, so it should favor their analysis. So here is the mean of the traits. Sigma is the covariance of the phenotypes. Okay, so it captures the correlation structure of the phenotypes. And this matrix captures the correlation between samples, among samples. Okay. So basically, this simulation is not assuming independent samples, even though IHD assumes independent samples. Okay, so we do not model this here. And when we did a simulation, we tested small number of traits like 2, 3, 5, and then large number of traits, 10, 50, 100. Okay. And here is the simulated result for 2, 3, and 5 traits. We compared the multivariate version of IHT to the univariate version. Okay, so multiple univariate GWAS. And then we also compared it to CCA, which is multivariate Plink. And this is the second fastest software. And multivariate linear mixed model is the Gamma software. Okay, so in terms of time, we are quite fast, right? Even for as long as there's 3 traits, IHD is faster than CCA. And for 5 traits, IHD is like 5 times faster than linear mixed model. And this, by the way, this timing includes cross-validation time. And then we also looked at the statistical performance here. The pleiotropic true positive is the true positive rate of pleiotropic SNPs. And independent true positive is the true positive rate of independent SNPs. Okay, basically, IHD has similar true positive rate compared to Gamma. And for the false positive rate, IHD actually is much better than all other methods, right? Sometimes 10 or 100 times better than these other methods. And multivariate linear mixed models has fewer false positives than canonical correlation analysis, as it should, right? This is the main reason they tout their statistical performance. So if we increase the number of traits, right, to 10, 50, and 100, this is the result. Basically, Gamma will not run for more than 10 traits, right? For 10 traits, our IHD is already like 20, 30 times faster. For 50 traits, Gamma won't run. and we are 10 times faster than CCA. And the statistical performance remains, okay? So basically, IHT scales linearly in terms of the number of traits, right? From 50 to 100 traits is just doubling the time. So really, you can, if you want, you might be able to do like 500 or 1,000 of traits with IHT. So now we move on to the real data analysis, and this data is exactly the same as, the data featured in our first IHT project. So here the phenotype is systolic blood pressure, diastolic blood pressure, and BMI. Okay, there are about 200,000 samples, 500,000 SNPs, and this data would be 700 gigabytes if you decompress it to numeric matrix. Okay, so really you can't really do this naively unless you really want to, right? But usually you cannot do this. So we included these non-genetic covariates, and then we cross-validated multiple paths, and it turns out that 197 non-zero entries in beta. Now 197 non-zero SNPs is discovered, and doing all of this analysis took about 20 hours on 36 cores on a 36 core machine. So here's the result. The IHT found 13 SNPs that are pleiotropic, and he found 171 SNPs that are independent. So what we did was we took each of these SNPs and then we searched a 1 megabase region flanking them for other associations in the GWAS catalog. And it turns out that all 13 SNPs, all 13 pleiotropic SNPs were already known, and over 90% of independent SNPs were previously known. Okay, and we did find 10 new associations with diastolic blood pressure and 3 new associations with systolic blood pressure. And we call them new because they are not within 1 megabase region of any known SNP associated with these traits. It also turns out that our software estimates the correlation of the traits, and it is here. And we can also estimate the proportion of genotypic variants explained for each trait. Okay, and it is listed here. And they are underestimations of heritability for these traits, right? We call them chip heritability. I don't know, right?
Sorry,
So
what
these
kind of heritability?
Chip heritability.
Chip.
I don't know. It's not heritability.
I had another question. The 13 pleiotropic SNPs that you found, and you're saying they're known, were they known as pleiotropic for the same sets of traits that the diastolic and systolic and BMI? Were they just known for one of the 3?
They were known for at least one of the 3. So I didn't check whether they were known as pleiotropic.
Right.
I don't know how to check that.
Well, see, they're known individually for more than one, but sure.
So this is the main result we have. So here's a summary for our last project. So we compared the multivariate IHT against Gemma's linear mix model and against multivariate Planck's canonical correlation analysis. And we have similar true positive rate, much better controlled false positive rate, and our algorithm scales linearly as the number of traits increases. And importantly, IHT is the only software that will run for more than 50 traits simultaneously. And to demonstrate the scalability of our software, we applied it to UK Biobank with three traits, right? And it took less than a day on a single machine. And more than 90% of SNPs discovered were previously known, right? So it reproduces previous results. So here's the overall conclusion. So how did we deal with the special structure of the phenotypes Y, right? For the univariate case, we assumed independent samples and performed maximum likelihood estimation under generalized linear models. Okay, so that's how we respect the structure of Y. How do we deal with missing genotypes? Or I guess I should say, in order to impute these missing genotypes, it is very slow, right? So we developed some very fast phasing and imputation algorithm. And it turns out that that will allow us to partially deal with the population structure of data. And what about the challenges of big data? We do have some routine to work with compressed genotype matrices, and that will allow us to save 30 times memory, but computational speed is competitive with BLAS, with double precision matrix multiplications. How do we incorporate external evidence? We did not talk about this, but IHT can do like group projections and weightings, just like how you do group lasso and weighted lasso. And we just didn't show it here, but we can do it. And what do you do when you have multiple phenotypes, right? We assume independent samples and then use the multivariate Gaussian to model the correlation. Okay. So finally, this is, I guess I want to just thank my committee members, especially my advisor, Ken and Janet. They have been extremely helpful this entire time. And then my committee members, Hua and Eric, they also were very heavily involved in all of my projects. So I really thank them as well. So that's it. I would like, maybe I can take questions if there are any. Okay.
Wait. Wait. Then I had a quick question, I think. If you can go back to your results, I'm curious about one thing. This is your final project. Yeah, that nice your simulated results, sorry.
Oh.
Your nice table. Okay, you can stop at this one. This would be a good one to stop. I was interested in those, when you show the first set, I'm like, oh, well, maybe I should just use the univariate IHT because it doesn't seem that's much of an advantage of the multivariate. However, it looks like as you increase the number of traits, the false positive rate for the univariate starts to climb up relative to the multivariate. Do you have any insights to maybe why that is? Is that
Why multivariate has better false positive rate for?
Yeah, potentially. So I'm just trying to think, yeah.
I guess for univariate IHT, it has to run 100 separate analysis, right? So it has more chance of finding false signals. I don't know.
Yeah, I'm curious. Yeah, I don't know either. It was interesting because it doesn't, as I said, it seems to be, and it may be, I mean, this is just one realization and things, but it was interesting because when you looked at if you go back one other step, right, it doesn't seem that was happening.
Yeah.
So they look more comparable. So I was just curious if you had any insights. It looks like, am I right that the multivariate IHT is a little bit better about picking up the true positives than the univariate? Is that right? Yeah.
Seems like yes, or small number of traits at least. That seems to be true.
Okay.
Although not much better, just slightly better.
Yeah. Okay, thanks.
Yeah. And I think, is that the slide before this one? That's a simulating model, generative model. So it's interesting, in the generative model, you do have the GRM, the kinship there.
Yes.
I mean, usually people argue like for GWAS, if there's lots of like a subpopulation structure, you should use linear mixed model like Jamba to adjust for that. But in the simulation result, actually Jamba has more false positives than your method. Your method is assuming independence, right?
Yes.
I think it's interesting.
But I did choose a small sigma G here. So heritability is like 10%. I don't know. If
yes, here 10%, but you didn't adjust for that at all.
Yeah.
And it turns out the method which adjusts for that, which is Jamba, has more false positive, which is that true? Yeah, I mean, if it's true, it's interesting.
But you know, these false positives, they are basically picking up a bunch of SNPs that are in LD. With the,
oh, got you. I see.
So it's hard to say they are
right.
false positive.
Right. I see.
But even then, IHD does better, right? So I don't know.
So
In
it's
practice,
safe to say, Ben, among the SNPs that are an LD, it's picking out the right one that shows the strongest signal with the trait.
Yeah.
And for marginal approach like Jamba, you use just genome-wide significance level. Is that right?
Yeah.
I see.
Not genome-wide, the bumper only. So 0.05 divided by this.
Okay, I see.
Are there any questions from the audience?
I have one question about your implementation, Ben.
Yeah.
On this project. Since you mentioned that you have small matrix inverses, I was wondering if you tried using static arrays to handle the matrix inverses.
That is a to-do list.
Okay, yeah, I was curious because that you might see an even greater speed up from just that.
But this is a very small. It takes 0.1% of the whole time.
Right, so it wouldn't be a huge impact on performance. Yeah. I see.
Overall time, this is very tiny.
I see, thanks.
Ben, I have a quick question. The 3 slides, the matrix normal slide.
Yeah.
Like given this, can we calculate, like given the assumption we can't calculate co-heritability between, like, give any pair of traits, is it simple enough or under your framework?
Calculate
co-heritability.
I think that requires a variance component model, which is not the model Ben's.
I see. Okay.
Under the simulated model, the covariances are Kronecker products, entries in Kronecker products.
Oh, I see. Right. So is that if without the sparsity constraint, the objective function is convex, right? Where it's concave, you are maximizing. Is that right?
Is it?
Looks like so. That means, well, is that the reason you choose the precision matrix as the argument parameterized in terms of precision?
I don't know, just because we are working with, we don't want to work with, update the inverse of a matrix, right?
It makes some of the derivations a little simpler, Wah, because you're not differentiating the inverse of a matrix.
Right, right.
Either way, I think is acceptable.
Then is it easy to extend to nonlinear traits, you think?
Sarah's
Like-
working on that. So
Mm.
eventually it'll be done, probably. Yeah, she'll do the quasi-copula thing, right? It will work with different non-linear traits. And even if it's like, you know, one of them is binary, one of them is count, she will do it eventually.
Right, with coupler, right.
But not with IHT. I think that might be hard.
Mm-hmm. I see.
Just another quick question about your simulated data set.
Yeah.
The Northern Finland birth cohort. I mean, does that? So the genotype samples are directly from that. You didn't simulate the genotypes, right? Only the trait values?
Yeah, only Y is simulated.
Yeah,
Oh, and sigma.
right. Is there much, I mean, what does the GRM roughly look like? Is there a lot of close relationships amongst the NFBC?
Oh,
Are there?
I don't know. I never looked.
Sure. I mean, if they aren't actually close genetically, then maybe that explains why the GEMMA doesn't do much better. If they aren't actually primary or secondary relationships.
Maybe, right? Maybe.
But Gemma has the advantage, Eric, of trying to account for those relationships.
Right. But if they don't actually exist in this particular data set.
Oh, yeah, then they're kind of on a par.
Right.
But I think this data, they do have.
Yeah,
I don't know. Usually Finland, I think the paper said they are more homogeneous than normal.
Certainly there is some, I mean, that they're related somewhat, but maybe distantly, I don't know.
I see. I don't know then. Maybe maybe that's why.
If there are no further questions, let us congratulate Ben.
I have one more question.
Oh, good.
Ben, can you go to the last slide? So I'm wondering, I mean, it's crazy. I mean, you can like do R like greater than equal to 50 or 100.
Oh, yes.
What kind of applications you can requires fitting this kind of many phenotypes together? Like EQTL or something?
Yeah. And then I think there's a bunch of like other non-GWAS stuff, right? The PWAS, whatever. I don't know.
Yeah, I just wonder you should try to explore it a little bit more, so it has a more higher impact. It can be used in some crazy R applications. You know? Yeah.
But you can imagine why in an EWAS, a similar SNP might control many genes. So you would pick up a lot of pleiotropy.
So that's EQTL type.
Yeah.
Yes, I see. Yeah.
But the EQTL, the phenotype is count, right? So can you still do that?
Microarray is continuous. If RNA-seq, it's count.
Yeah. Yeah. And, you know, a lot of times you can get away with some kind of transformation that is reasonable.
Mhm.
So I think you could look at some things, but you're right. I think a lot of the FEWAS type of things too, a lot of biobank kind of data would have hundreds of traits. I don't know, Sriram, are you still on here? If he's still here, I don't know if he is. He's got some nice applications, I think, where they look at lots of traits. It looks like he's gone. So
Yeah, UK Biobank released metabolic traits. Those are all continuous.
yeah,
Yeah, and potential apply.
Yeah. Metabolome type things would be
Yeah.
Another example. I mean, you can easily go exceed thousands of traits if you really want to.
Mhm.
So
That will still take a long time though, because
mm,
this is only one chromosome. So
yeah. But you know, the fact that you have NAs on many of those is interesting. And maybe, you know, when you start getting into that kind of thing, maybe it's useful to try to do some of the univariate stuff first, screen out a bunch of stuff that really doesn't look relevant, and then do the multivariate on a subset or something like that.
Yeah, Ben, on this 100 trait example, this result is about mapping, gene mapping. I wonder, did you look at the estimate of the precision compared to the truth? How I'm a little worried, like 100 by 100 matrix, that's like thousands of parameters. Your
Yeah.
sample size, what's your sample size?
it's 5,000.
Yeah, I'm worried. I mean, the estimates for the, you know, covariance estimation, it's hard. I mean, it's 100 by 100. I wonder, did you see the estimates for precision? How do they compare to the truth? The 100 trait example.
I didn't look at it. I didn't look at the 100 traits,
Uh-huh.
but I wasn't sure how do I compare them? Just
Well,
for-
this is a simulating example.
Yeah.
Just find the estimates
just the difference and then
compared to the truth. Yeah.
Oh, I didn't look for 100 traits. Well, maybe
I think it would be fairly easy. And Eric Chi and I wrote a paper a few years ago on putting Bayesian priors on estimation covariance matrices, and it works pretty well.
Yeah, put sparsity on precision. Yeah.
Well, not sparsity, but decent condition number.
Oh, okay. I see.
You could try sparsity too, for that matter.
Then you get some, if it's a EQTL, you get some network, gene network. You can, you know, that's graphical model essentially if you put sparsity on precision, right?
That's right.
Yeah.
That's interesting.
You may recover some gene network. Yeah.
What this is telling me is there are many directions Ben could take his work.
Right.
So is it time for us to excuse the rest of the audience and have a quick committee meeting with Ben?
I suppose so, Janet.
Okay. It won't be long. So those of you who are having a party, stay tuned.
thank you all for coming.
I don't have any further questions.
No, we should just go a little bit over procedure, but Ben, that was a very nice talk. I really enjoyed it. I thought you covered things very well. So I have no further questions either.
Thank you.
Yeah, I don't have further questions as well.
So Ken and I talked with Ben on Friday, and I told Ben I didn't have any other suggestions or changes for his dissertation.